Hello, dear students. In this lecture, we'll discuss about the another fourth test in IMV classification of indicator organism which are present in the water. On that basis, we can determine whether that sample is contaminated with the fecal organism or not. The fourth test, that is nothing but the citrate utilization test. So test already itself suggests whether the organism's ability to utilize citrate as a sole source of carbon. It completely depends on that particular mechanism. In this test, this test is used to assess the identification of enterobacteria or the organism which are belong to the Enterobacteriaceae family. And this test is used to determine the ability of the organism, or we can say bacteria, to use citrate which is present in that particular media as a sole source of carbon and energy. So for that we can use the media which contains citrate as a carbon source in that particular media. The ability of that organism, it is completely depend on the presence of citrate permease. It is nothing but the enzyme which is synthesized by that particular organism, which facilitate the transport of citrate in the cell. So here, if the citrate permease is present, so it facilitates the transport of citrate which is present in that media into the bacterial cell. Once inside the cell, citrate is acted upon by the enzyme citrase and dissimulate the sodium citrate. So if the citrate is enter into the cell, it acts upon the enzyme citrase and it forms a sodium citrate which is present. Sodium is already present in that media. During this reaction, the medium becomes alkaline, and that, uh, as we can see, if the media becomes alkaline, that is nothing but the change in the pH of that particular media, and on that basis, the color of that media can be changed, and we have to use that particular change in color in that test for the detection whether that organism has ability to utilize citrate as a sole source of carbon. During this reaction, the media uh, which becomes alkaline, the carbon dioxide which is generated during the dissimulation, which combines with the sodium and water to form a sodium carbonate, which is alkaline end product of that particular reaction, and can due to that the color of the media can be changed. This alkaline product which increase the pH of that particular media and change the bromothiamol blue, which is indicator used in that particular media or the medium, from green color to deep blue in color and represent a positive citrate test. So in that case, we can normally use the bromothiamol blue is nothing but the indicator in that particular media. In case of that, as we can normally prepare the media at neutral pH, and as the pH of the media change to into alkaline pH, the bromothiamol blue, which is already in green in color, which uh, change in color can be observed, and that becomes a deep blue, or so in other words we can say it is nothing but the it becomes a blue color media if the organism has ability to synthesize citrate as a source of carbon. And it is nothing but the basically if the organism has a citrate permease enzyme, so that the citrate which is present in that particular media, it facilitate the transport of citrate inside the cell, and due to that the uh, it end product of the reaction is nothing but the sodium carbonate, and it is nothing but the alkaline media, alkaline nature of that particular end product, so that pH of the media can be changed. In this test, the test organism are grown in a Siemens citrate agar. It is nothing but the media Siemens citrate used for the inoculation of organism, and the procedure is nothing but the similar. Only the difference is that in case of indole and MRVP, there is a broth media we can use. In case of citrate utilization test, we use a solid media. Uh, it is nothing but the agar medium, which contains sodium citrate. It is nothing but the carbon source, and ammonium sulfate as nothing but the nitrogen source, and bromothiamol blue as a pH indicator. On that basis, we can determine whether the end product is synthesized in that particular reaction or not. So in this reaction, we can see nothing but the if the organism has the citrate lyase enzyme which break down the citrate and it forms a oxaloacetate and acetate. After the conversion of the oxaloacetate, oxaloacetate decarboxylase enzyme, it forms a pyruvate and carbon dioxide. In case of acidic condition, that pyruvate it forms acetone plus lactate and carbon dioxide is released. In case of alkaline condition, pyruvate it forms acetate plus formate and carbon dioxide is released. So completely the reaction is depend upon the acidic and alkaline pH of that particular media. If the carbon dioxide it bind with the sodium, it forms a sodium carbonate and it is an alkaline product. And due to that, the pH of the color of the media can be changed. Bromothermal blue, which is indicator, it present in that particular media. In that diagram, we can see it is nothing but the basically media which is prepared before inoculation, and it is nothing but the blue in color. uh, Green in color, as bromothermal blue is nothing but the indicator which is used in that particular media. Bromothermal blue is yellow at acidic condition and green at neutral, and blue at is become in case of alkaline pH. So here in the diagram we can see if the organism has ability to utilize citrate as a sole source of carbon, the color of the media becomes blue, and in the case of that media remain as it is considered as negative taste, and if it becomes yellow, we can say end product is nothing but the acidic. So the pH of the medium initially adjusted at nothing but the neutral pH. The indicator gives green color to the medium. As the taste is negative, the media remains as it is in the form of green color. In order to perform the test, the organism which can see we can call is nothing but the experimental organism is inoculated on Siemens citrate agar, which is a slant medium, and after inoculation, the slant are incubated for 24 hours at 37 degrees Celsius in the incubator, and we have to observe after 24 hours for the change in color. If the color change is observed, we can say it nothing but the organism has ability to utilize citrate as sole source of carbon, or on the basis of changing color, we can also determine the organism is able to utilize citrate as a source of carbon. The color change of indicator from green to blue indicates the positive taste. If it remains as it is, we can say it is nothing but the negative taste. So it is nothing but the principle behind citrate utilization test. In that regard, we have also used the uh, four tests, that is indole test, methyl red test, VP test, and citrate utilization test. And that tests are used to differentiate fecal and non-fecal organism into different organisms. So on the basis of immune classification, we can see it is nothing but the uh, table. On that basis immune classification can differentiate. Here is nothing but the organism we can consider they are from the fecal origin or non-fecal origin. Here is the report for the indole test, methyl red test, VP test, citrate test, and on that basis that organisms are considered to be whether they are from fecal or non-fecal origin. If the E. coli variant first is found in that particular media, it gives indole test positive, methyl red positive, while VP and citrate utilization test is negative. So that considered to be the organism from fecal origin. If E. coli variant second or another variant is present in the media, it gives indole negative, methyl red positive, VP negative, and citrate utilization test. And also we can say it is nothing but the organism from the fecal origin. In case of Escherichia profundi and Enterobacter erogens, the both Enterobacter erogens are normally you can use differentiate between fecal and non-fecal coliform. So it gives indole test negative, methyl red negative, while VP and citrate utilization test positive. And both the first E. coli and Enterobacter erogens normally you can use whether they are from fecal origin and from non-fecal origin. Apart from that, also Escherichia sorry, another organism we can also use which gives indole test negative, methyl red test positive, VP test negative, and citrate utilization test positive. So basically, the first and last organism we can use whether first organism gives two test positive, two test negative, that is positive, positive, negative, negative, and interbacterial it is nothing but the negative, negative, positive, positive. So on that basis, we can differentiate the whether that water sample is contaminated with the fecal coliforms or non-fecal coliforms. So there is nothing but a differentiating test we can use to differentiate organism between fecal and non-fecal origin. Thank you.